Right. Thank you. Tinggal How was the week? Good. I'm thinking I'm waiting. Anh ấy nghĩ là sang kỹ lên đấy ấy xin rồi mà những cái bị đánh thao thì phải là මේකේ ෆස්ට් පාර්ට් එක පටන් ගන්නකොට ඒගොල්ලෝ කියන්නේ නේද? ඒත් වාගේ ටයිම් එකේ Right. This we started last week, one before the last week. Well, we discussed about blood sugar test.
Under that, we had several categories: fasting blood sugar, random blood sugar glucose, and OGTT. In the test, we talked about how to collect the sample and the clinical side and the reference ranges, how to do the calculations. We talked about BUN test, blood urea nitrogen test. Creatinine test. කරාද කරානේ Right, okay. This is where we have to start from. Cholesterol. Right, we learned about cholesterol and the lipids. Right, I think lipids and another place also we made cholesterol up to some extent. So officially under lipids we talked about cholesterol. So cholesterol test. First of all, if I remind you, cholesterol is a waxy fat-like substance, right? When we say cholesterol, we get a very bad feeling about cholesterol. But no, cholesterol is something that we need, right? It is essential for cell membrane structure and some hormone production, bile acid production, right? And it is transported in blood, right? Okay, so those are the general things about cholesterol. Well, right. Well, speaking of the cholesterol test, so what are the imports of that test? Well, we know cardiovascular risk. What is cardiovascular risk? You know, when it comes to the blood veins and arteries, the inner radius, right? Inner radius. If cholesterol gets deposited, the inner radius decreases. Then the tube becomes tiny. Then the blood pressure increases. The enough blood is not supplied to the certain parts of the body. And when that happens to the brain, then brain becomes malfunctioned because there's no enough blood, enough oxygen, right? Okay, so this is the risk that we have. We call it cardiovascular risk, right? Well, so that has to be diagnosed as early as possible. That has to be diagnosed, right? And also, when we diagnose that, then we can go for treatments. You know, when it comes to certain situations, like, you know, for everything in such situations, we have the stage 1, stage 2, stage 3, right? If we can recognize something at very beginning, stage 1, that is a very good thing. Then we can assess them basically. So in risk assessment, this is what we call risk assessment. Then as early as possible, we can diagnose this, right? And then risk can be evaluated. Then we can decide on the treatments, right? Otherwise, you know, when it comes to stage 2, stage 3, and stage 4, like that, it is very difficult to deal with it. Then sometimes there's a situation you might already know about, heard about these stuff like we have to give medicine to reduce the thickness of the blood thickness and බෙහෙත් දෙනවා කියන්නේ. ඇයි දැන් අර thick blood flow වෙන්න තරම් අර tubes වල තියෙන thick ඒ කියන්නේ දානකම radius එක මදි. Right? We might feel well. Bacillus will feel well. Right? So then that has to be assessed. Likewise, in such cases, you know, treatments also should be done in steps. First, we have to reduce thickness of blood because at the first place, blood has to be supplied. The thick liquids, blood is a thick liquid that doesn't go through very tiny tubes. when the radius decreases, so then that has to be assisted. That's important, right? Okay. And also, with the test, then we can have an idea about the last point I'm talking about. It evaluates the liver function and fat metabolism. Because when it comes to cholesterol, fat, the lipid related things, it is the liver which is playing the major role of that. Right? So then we can have an idea about how functioning the liver is. That is also important. Okay, right. Right. Then, when it comes to blood tests, we have several categories. Likewise, when it comes to cholesterol test also, there are several categories. Right. We go for total cholesterol. Total cholesterol. Blood test වගේ තමයි හොඳ. අර අපි ගන්න full blood count. Fasting blood sugar, normal heat. In full blood count වගේ some of so the total cholesterol. Right? We measure the total cholesterol. Right. That means it is the addition of collection of all the cholesterol components. What are the cholesterol components කියන්නේ? cholesterol විදිහටම තියෙනවා තියෙන්නේ. ඒ වගේම තමයි cholesterol තව compounds එක combine වෙලා තව ඊට වඩා complex compounds හදනවා. අර ඒව තමයි අර thick waxy layer which is going to be deposited on the inner wall of the arteries. Right? So then collection of all the cholesterol compounds. That is what we call total cholesterol. Okay. Take a testing. Another one. Right. HDL cholesterol, high density lipoprotein. We call them as good cholesterol. Good cholesterol. Right. So another category. We can measure the amount of good cholesterol, HDL content. That means like if we get a count about HDL count, then automatically, proportionately, if the HDL count is high, the other bad cholesterol count is going to be low. Then, right, that is that. And the other version, LDL cholesterol, low density lipoprotein. We talked about this category also before. We have two types as good cholesterol and bad cholesterol. So then low density is coming under bad cholesterol. Right? Okay. Then we do the test for finding out how much bad cholesterol is present. That's the other test. And the other one, VLDL cholesterol, that is very low density lipoprotein. So that is also considered as bad cholesterol. HDL is good. Right. And we have triglycerides. Right. So these are the measurements. These are the things that we measure in cholesterol testing. Right. When you do this test, the sample that we collect from the blood, from the body, that is either serum or plasma. Then if you go for a fasting sample, that is going to be 8 to 12 hours, right? May be required for accurate triglyceride and LDL values because HDL, of course, it is good enough, right? Then more than HDL count, what is important is LDL count, right? Have you done a cholesterol test at least once? No. Right. So when it comes to like 40 වගේ වෙද්දි අනිවාර්යෙන් කරන්න. And that also only if you are active. You get in කාර් එකේ ගිහිල්ලා කාර් එකෙන් බහලා ගිය ඇතුළට ගිහිල්ලා ආයෙ කාර් එකේ ගිහිල්ලා ඊළඟ සෙන්ටර් ක්ලාස් එකේ ගිහිල්ලා කාර් එකෙන් බැලුවල ඒ උකට තමයි ඇක්ටිව් කියන්නේ. Right? At least පොඩ්ඩක් ඇවිදිනවා නම් පරිගලක් නගිනවා නම් ඒ මිනිමම් දේවල් ටික හරි තියෙන්න ඕන. Right? So in such cases, in the blood circulation rate is going to be high. Or monkey or the vaccinator or deposit. It is estimated that half an hour walk කරන්න ඕනේ එහෙම අපි consider the පොඩ්ඩක් විතර ඒක මදි because that is coming under the warm up. ජිම් ගිහිල්ලා ඔකනේ අපි ඉස්සලා ට්‍රේඩ් මීල් බැදලා මේ ටික කරලා නේ ඒ warm up. Right? Okay. ඊට පස්සේ දැන් අපි daily restaurants වලින් කන්න උද්දක් විදියට කන්න ගියාම cholesterol level එක වැඩි වෙනවා. That also should be considered. Right. Okay. So when we do the test, the reference range, right? If we go for total cholesterol count, it has to be less than 200 milligram per deciliter, right? And HDL that changes with the gender, right? For men, it will be greater than 40 milligram. For women, it will be greater than 50 milligram, right? That is that. And LDL count, right? LDL count, that's bad cholesterol. Bad cholesterol should be lower as much as possible, low, right? So that is going to be less than 100, right? Less than 100. Triglycerides, it should be less than 150. Okay, so this is going to be the reference ranges. When we do the test, our value should be within these ranges for us to consider that we are healthy. Well, so what exactly we do during this test? We collect sample, serum, okay, we collect sample. After we collect the sample, this is how we going to do. We add cholesterol esterase. That is an enzyme, right? Cholesterol esterase. That converts. Just give me a second. Well, what I was talking about was, right, the enzyme cholesterol esterase, right? It converts cholesterol as like it is converting this into free cholesterol. That is step number one, right? That is done by an enzyme, cholesterol esterase. Then we have free cholesterol after that. In the second step, what is happening? Cholesterol oxidase, right? That converts cholesterol into cholesterol 3 on and hydrogen peroxide, right? Now we have hydrogen peroxide. Then peroxidase that reacts with hydrogen peroxide and a dye, it forms a colored complex. It forms a colored complex. Then what do we do? We measure the color intensity, right? We measure the color intensity. Let me verify. We have the sample serum, right? We add cholesterol esterase, right? That converts that into free cholesterol. Step number one, right? What do you mean by free cholesterol? Cholesterol, it's in compounds, it's combined with other substances, right? Right. So then all the cholesterol versions are converted into free cholesterol by the enzyme called cholesterol esterase, right? And then cholesterol, then cholesterol oxidase that converts the free cholesterol into cholesterol 3 on and hydrogen peroxide. Now we have hydrogen peroxide. That hydrogen peroxide, peroxidase reacts with that hydrogen peroxide, will form hydrogen peroxide, and a dye forms a colored complex. දැන් එතකොට cholesterol අපි ගත්ත sample එකේ cholesterol ගොඩාක් තිබුණා. හැදෙන color complex එක color substance එකේ ඒ color intensity එකත් ගොඩක් තියෙනවා. නේද? dark color එකක් තියෙනවා. cholesterol අඩු නම් අර color intensity එක අඩු. right. ඔන්න ඔය color intensity එක තමයි අපි බලන්නේ cholesterol කොච්චර තියෙනවද කියලා. right? Okay, so then we finally measure the color intensity. This is how this happens. That is the method, basically, the concept. For us to do that, we need these substances. Cholesterol esterase we need for the first step. For the second step, we need cholesterol oxidase. For the third step, for reacting with hydrogen peroxide, we need peroxidase. Right. Here was our color region because either right. So chromogen. Right. So those are the things that we need. Then we need a control experiment. That is cholesterol standard cholesterol solution is needed. That is to compare with. And we need distilled water. I think you remember these three solutions, like we have three plastic also we were talking about. What is that? We have blank solution that is just with distilled water. And we have a known solution of the particular substance. Cholesterol, but not cholesterol, අපි දන්න solution එකක්. දන්න concentration එක, දන්න amount එකක් තියෙන solution එකක්. ඊට පස්සේ ඒ second එක. Third එක අපේ patient ගේ sample. නේද? ඔය තුන තියෙන්නේ. Right. Then we have those three test tubes. Okay. Requirement of sample. We need serum or plasma sample, right? And collection plate or gel tubes, right? 8 to 12 hours fasting for lipid profile, right? Uh, These are the requirement about the sample. Reagents we discussed about previous slide. And the sample, these are the conditions about sample. That is from the patient. Right. The blank one, we have the reagent. Blank one, standard one, test. Test is the patient sample. We have the reagent. And also, so then we have distilled water for the blank one and cholesterol standard, non-cholesterol sample to the standard one. And the test is, that is the patient's serum sample. Those are the three. Then we have three test tubes. Right. We label those. We label the three tubes. Then we add, by using a pipette, we add 1 milliliter, 1 centimeter cube of the reagent. Right. Of working reagent. That means working reagent means when it comes to these reagents, there is an expiry date for that. The test will not be valid after that. Then working reagent means the reagent which is in the active state, in the usable state, right? Then we add 10 microliter of distilled water, standard or zero, right? Those three. Then we do the incubation part. I think we were talking about what incubation is in our previous session, right? This is for 10 minutes, right? So 13.7 degrees. So room temperature for 15 to 20 minutes. Right. That is incubation. Right. Then we measure the absorbance. Absorbance is the color intensity. Definition. This is the calculation. This is the absorbance of our test. That means patient's sample divided by standard solution, that is non-cholesterol samples color intensity into the concentration, right? So if you look at this example, so let's say the patient's value is going to be 0.6 and the standard value is going to be 0.5 and the concentration of the standard solution is 200, then we get the final answer 240. That is how we get the final answer. That we check with the reference ranges. Clear? That we check with the reference ranges. That is how we get the final answer. Right. As always, during this bioclinical test, the quality of the test has to be definitely maintained. When the quality is not maintained properly, we can't rely on the figures that we get. That has to be done. So, what do we do? Run normal and abnormal cholesterol control serum with each batch, right? So the normal and abnormal are both. Okay, right. And also, calibration has to be done properly. What is calibration? Now we are using a spectrometer to check the color intensity. The color intensity has to be like the instrument should be working properly, right? So then that is another one to control the quality of the test. Well, during this, we have to be careful about certain things. That means we have to avoid certain things. So make sure that our experiment is properly done, and the result is reliable. Well, avoid hemolysis. What is hemolysis? You know, hemoglobin, if hemoglobin is present in the medium, obviously hemoglobin interferes with the color of the reaction, right? We measure the color intensity. Hemoglobin itself, it has the color, the red color, isn't it so? Then that interferes with the color intensity that we measure, right? Okay. And the region has to be, the proper regions has to be used. And timing for accuracy, that is to be ensured. And when we measure the solutions, we have to prepare out the right amounts of solutions. Well, so these are the precautions that we must take during cholesterol test. For this test to be considered as a valid test and reliable result, what are the things that we should consider? Right. Increased total cholesterol. Well, we call it hyperlipidemia. Right. Then, of course, so then when we evaluate the value, the cholesterol level, the lipid level, right? So it is high. Well, that means we have high level of cholesterol, high level of triglycerides in the blood. That problem is there. Well, and the other thing is, other situation. So then what we are talking about right now is where after we read the report, we read the report now. After reading the cholesterol report, what conclusions are we coming? Right? They will say the report is not satisfactory. That means that our values are going to be bad in such cases. Right? Okay, so then cholesterol will be high. And the other thing is hypothyroidism. What is that? Well, that is something related to thyroid gland, which we saw before under hormones, not very recently, right? Then what is happening is that the thyroid gland is not making the required amount of thyroid hormones, which our body needs to function properly. That is not happening in a sense like. So after we read the report, the ideas that we get, right? And there's another situation. Well, think about a situation where from the kidneys, the protein is getting leaked out to urine. You might have, it is not, it is some type like, it's just situation, health issue that exists in society. Right, you might have heard about that. Protein is getting leaked to urine. That means urine is detected when we go for a test for urine test, basically. Urine test. Urine test indicates that proteins are present in urine, right? Then there is something wrong with the liver function because when it is filtering properly, not properly actually, when it's filtering, the proteins are leaking out to urine. That problem is there. Right. Then with cholesterol report, that also can be considered. Right. Diabetes situation. Right. A diabetes situation. That is also can be developed. And the last point is about LDL and stuff. low density lipoprotein. And the LDL level is high. That leads to heart diseases and strokes. So those are there. Those are there. These are the concerns that we have after reading the report. We were talking about the increased level of cholesterol. What about decreased situation? That is also there. Decreased cholesterol. One thing is the liver is not functioning properly. That is possible. Another thing is malabsorption or malnutrition. Malabsorption, malnutrition, what is the difference between those two as you think? Malnourished and malabsorbed. Malnourished කියලා කියන්නේ අපි කනවා. නෑ අපි කන්නේ. හරිද? ඒ කියන්නේ කනවා හැබැයි healthy නෑ. ඒ කියන්නේ හරි කනවා. But the required nutritions are not there. හරි. malnourished. හැබැයි malabsorption කියන්නේ ඒක නෙමෙයි. අරකට ගන්න නෑනේ. malabsorption කියන්නේ ඒක නෙමෙයි. අපි කනවා. හැබැයි absorb වෙන්නේ අවසාන ප්‍රතිඵලය ගත්තට පස්සේ දෙක වෙනයි. දැන් කන්නේ නැතුව අපි මන්ද පෝෂණය හැදෙන එකයි. කාලත් මන්ද පෝෂණය හැදෙන එකයි කියන්නේ ඒකේ දැන් දෙවිදියක් වුණාට අවසාන ප්‍රතිඵලය එකයි. හරි. එතකොට so malabsorption ඉතින් malabsorption පාඩම් කියන්නේ ඒක තමයි. ඉන්නේ මොකක්ද නෑ. ඔය කරන ප්‍රමාණයක් හරි. එතකොට malabsorption වලට මම එක්සැම්පල් එකක් දෙන්න. දැන් නිකන් malabsorption දැන් අපි සමහර වෙලාවල් කියනවා අපි සමහර කෑම වලින් පස්සේ තව මොන හරි කෑමක් කන එක සුදුසු නෑ කියලා අපි සම්ප්‍රදාය ඇතුලේ අහලා තියෙනවා. ඒ මොනවාද? එක්සැම්පල් එකක් දෙන්න. absorption එක අඩු කරන ඒවා කියනවා. absorption එක වැඩි කරන ඒවා කියනවා. මොනවාද කියනවා? හරියට හරි. කාලා කොච්චර වෙලාවකින් පස්සේ තියෙද්දිවත් healthy 2 hours. එතකොට ඒකෙදි absorption අඩු කරන්නේ මොනවාගේද? මොකක්ද absorb නොවෙන්නේ? කාලා තියෙන්නේ. යකඩ. එතකොට iron absorption එක අඩු වෙනවා අපි දෙන්නේ. right? මේක පැය දෙකක් යනකම් තේරුන්නේ නැහැ. iron absorption එක වැඩි කරන්න පුළුවන් කරන්නේ කියන්නේ. ඒ කියන්නේ කාලා බේබීලා ඒක කම්පන්සේට් කරන්න තව මොකක් හරි ගන්න එක නෙමෙයි. දැන් කාලා බේබීලා sorry ඒක කරන්න දෙයක් නැහැ. ඒක absorb වෙන්නේ නැහැ. අපි ලොකු බේබොන්නේ නැහැ. right? කාලා අපි iron absorption වැඩි කරන්න iron අනිත් ඒවා තමයි. iron absorption වැඩි කරන්න අපිට ගන්න පුළුවන් මොනවද කියලා අයන් මම අයන් කතා කරේ ඒක සාමාන්‍යයෙන් අපිට තියෙන ඩෙෆිෂන්සි ගත්තට පස්සේ අයන් ඩෙෆිෂන්සි තමයි නැවෙල් පුරුදි. ඒකයි. මොනවද අපි කරන්න පුළුවන් අයන් ඇබ්සෝර්බ්ෂන් වැඩි කරන්න. අයන් ටැබ්ලට් හරි කැප්සියුල් හරි අයන් අරන් කියනවාද කොහොමද? ෆාමසිටිකල් ඇප්ලිකේෂන්ස්. විටමින් C අන්න හරි. right? iron අපි absorption වැඩි කරන්න පුළුවන් vitamin C වලින්. එතකොට දැන් හිතන්න දැන් අපි මොකක් හරි කියනකොට iron අපි iron supplement එකක් විතර අපි ගන්නවා කියන්නේ පොසිවල් හරි මොන හරි multi vitamin එකක් කියලා හිතන්න. එතකොට අපි ඒකේ iron absorption වැඩි කරන්න ඒකත් එක්කම vitamin C එතකොට බොනවා. අර 500 mg වල තඩි පැන්ඩෝල් size එක නෙමෙයි. පොඩි 20 mg පොඩි එක. right? iron absorption වැඩි කරනවා. එතකොට මේක ටිකක් කම්ප්ලෙක්ස් කතාවක්. ඒ කියන්නේ දැන් අපි කෑවටම බෑ. සමහර අවස්ථා තියෙනවා අපි එකත් එක්ක ගන්න ජාතියින් ඉන්න absorption නැතුව. අන්න mal absorption. එහෙම නැත්නම් සමහර වෙලාවට කියනවා ඇත්තටම අපිට මොකක් හරි රෝගී තත්ත්වයක් තියෙනවා. health issue එකක් තියෙනවා. එහෙම අපි අවුල් මොකොත් කෑවෙන්නේ නැති වුණාට අර ටික absorb වෙන්නේ නැහැ. හරිද? ඔන්න ඒක තියෙනවා. ඉතින් එතකොට මේතෙන්ට relate වෙන්නේ mal absorption or mal nutrition. So that is the problem. So decreased level of cholesterol when we come to that level, right? And hyperthyroidism, opposite kalikata, then chronic infections and inflammation. So then those are the other issues related to that. So that also we can judge with the medical report, basically cholesterol report. Those are the medical concerns. Right. What are the boundaries in this cholesterol test? Well, if we take a sample in like non-fasting sample, right? If it's a non-fasting sample, then that can affect the LDL, triglyceride and LDL. Total but we can't take that affects the triglyceride level and the LDL, the low density lipoprotein level. Well, speaking of the low density lipoprotein level, and so then basically this is how we do the calculation. We subtract, so total cholesterol is there. We subtract HDL and triglyceride level divided by 5. This is the calculation for LDL count. Okay, right? Because when it comes to total cholesterol, it is the addition of HDL and the triglyceride level. Then if we need to find out the LDL level, we have to subtract HDL and the triglyceride level. And there's a formula for that. You don't need to go up to that extent. Why do we divide this by 5 and all? At this level, you don't need. So then we'll stick to this place, right? Higher calculation is not required. But we need to know up to this extent, we need to know, right? Then we can find LDL count. Right. As we already discussed, we can assess the cardiovascular risk by using cholesterol test. And this is actually done by using a full lipid profile. And as I always say, this has to be judged with the clinical history. එකපාරට ගන්න report එකක් පිටින්ම ඒක isolate කරලා ඒක මෙහෙමයි කියන්නේ. Right? So that's what you get with experience, right? So then you have to analyze with the situation. Sometimes every 3 months, a report has to be taken likewise. So clinical history is important. And also lifestyle definitely matters. Lifestyle definitely matters. That also should be considered, which means just imagine that the cholesterol level will say, that's our topic now. In the cholesterol level, let's say that, okay, for a certain period of time, you kept on eating a lot abnormally, right? Let's say now think about that you attended to a wedding in India or somewhere, like people got 7 days in on wedding, they got a right? So then in such case, like after such time, if you go for a cholesterol level, then of course the report will say, right, you have too much of cholesterol, right? So that is what we call lifestyle. Then that has to be analyzed. So then that is going to be different from the normal situation. Right. Then with one report, we can't go for a conclusion. We have to go for another report. Right. So that's the end of the story. So clinical tests, that is that. All right. Hope you uh, have a clear idea about that. Right. About clinical tests, is there anything which is not clear? No. Hold on a minute, bro. Right. Okay. Since that is done, so then now we are going to go for an activity. අපි බලමු කොච්චර දුරට මතක තියෙයිද කියලා. Right. Your exam will be scheduled very soon. So I will update you as soon as possible after discussing with the management. Then I am not planning to give questions on the topic that I gave for assignment, because in assignment you already have studied about that. I know that you can remember that. So that time not going to test again, right? So a paper will be prepared avoiding that, because that has been already tested. And so the date will be updated as soon as possible. I will discuss and update you. And it's going to be a 2 hours paper. Yes, and that comprises with section A and section B, MCQ and writing both. Okay, that's how it is going to be. Right, okay. Let's move on. Right. Okay. So you are answering, then I am giving the directions. That's how it's going to be. It's not listening, it's a workshop. Okay. Okay, tapos pinili exam clear ng to, hindi pa ini. Right. What are carbohydrates? What can you see? Aku udah nih, ada yang kian. Ada yang tinggal ya, kan? Yeah. Polymer. Carbohydrate high. And singular version, carbohydrate just being with the carbon, hydrogen, oxygen. Yeah. That comprises with carbon, hydrogen, oxygen. Right? Basically carbohydrate. Number one, it is an organic compound. Right? And the elements present carbon, hydrogen, oxygen. One of the main type of nutrients that we have, that we depend on, and that is going to be the major energy source, right? So when we say carbohydrates, this is the description that we are supposed to know, right? That's about carbons. When we say what are carbohydrates, main types of carbohydrates. Hmm? Yes, we have simple carbohydrates, complex carbohydrates, then we have monosaccharides, disaccharides, polysaccharides. Yeah. Basically, like in detail, if I say we have simple carbohydrates, that is sugars like glucose, fructose, sucrose, sugar types. Starch, we have complex carbohydrates, right? That is found in bread, pasta, potatoes, like that. And the other category is fibers, that is also there. It's a complex carbohydrate that the body cannot digest. Fibers. Okay. What is the job of carbohydrate? The main job of carbohydrates in the body. Hmm? Sorry? Providing energy. Yes, that is providing energy. Basically, in our body, carbohydrates are converted into glucose. Then glucose is going to be used as the energy source in the cells. But you remember glucose? Yeah, one thing is it's coming under the character of simple carbohydrates, sugar. And energy wise, actually, that is the one like finally which is making energy. Is it so? In the metabolism part, it's the main source of energy in the human body. I think the door failed or something. There's a JBL also here, right? With the mic. Right. We have simple carbohydrates and complex carbohydrates, no? How do they differ from each other? What's the difference between simple carbohydrates and complex carbohydrates? What do you remember? Ratio thing. Uh-huh. Uh-huh. Again, simple. What's complex? Uh-huh. Again, structure wise. Hmm. Yeah. Right. When it comes to simple ones, because of the reason that you said, it quickly digests, right? Ratio say body ratio. But when it comes to complex carbohydrates, it's very long chains. Basically, we have sugar units, right? Then metabolism wise, for us to gain energy from a complex carbohydrate, it takes time. ඒක දැන් අපි මොකක් හරි ඇත්ලටික් ඉවෙන්ට් එකක් ගත්තොත් තේරෙන්න. right ඉවෙන්ට් එකට කලින් ග්ලූකෝස් දෙනවනේ. හැබැයි පත්ති ගානක් බෙදලා දෙන්නේ නැහැනේ. ඉවෙන්ට් එකට කලින් ටක් දාලා කාලි ගන්න කියලා. මොකද ඒක කාඩ් ස්ටාර්ච් ඕක ඩයිජෙස්ට් වෙලා ඊට පස්සේ අන්තිමේදී ඕක ග්ලූකෝස් වලට කන්වර්ට් වෙලා එනකොට ඉවෙන්ට් එක ඉවර වෙලා හැන්දෑව තමයි ඒකෙන් එනර්ජි. It's a glucose in right. Okay, it's a simple carbohydrate, simple one that quickly digests. We know that fiber is something which is not digested, but fiber is important. Not digested, but important. Not digested means okay, so that means it is not giving us energy, but still that is important. So then, without being digested, but still important, then why is that? What do you think? Fiber. අපිට fiber කතා කරා ඉන්නකොට ගොඩක්. කරා නේ කවදා. ඒ වෙලාවේ නිින්දකින් හිටියොත් මේ වෙලාවේ මම නැහැ ඒක සාධාරණ නේද තියෙනවා. දැන් මේ රෝ එකම දැන් already එසෙ නම් නෙදී. Right. Fiber. It regulates the body of the use of sugar, keeps hunger and blood sugar in check and promotes healthy digestion. That is, though that is not digested, then you know, then how it is happening. Like okay, we eat now. After that, you know, then it is going through. So then, when the fibers are there, it is something like solid structure. So with these fibers, it helps us to take the food after you eat in the digestive tract properly, right? It avoids constipation situations. because the fiber, around fiber, then the food and digested, half digested food will be there, right? So that helps us to maintain that part. But in case, what do you discuss about whether carbohydrates are bad for us? Under what circumstances carbohydrate is going to be bad for us? Will it be bad? Hmm? High glucose level. Okay. As a result of that. Hmm? So we are talking about the diabetes situation and all. Right, right. So this is an energy source, it's a different story. But if we have overconsumption of carbohydrates, yeah, in that case we have situations. And in that case we have situations because then in that case there will be a high level of glucose in the blood. Then finally this will lead to diabetic situation. Right? So then at the first place, we can't say it is bad because definitely this is essential for energy. Without carbohydrates, we can't have energy. That should be there. But the problem is uh, that will be creating health situations, right? One is diabetes, the other one is obesity, right? Becoming fat, right? So that problem is there. Okay. Where do you get carbohydrates? What foods are high in healthy carbohydrates? That is easy. That's about healthy carbohydrates. That means, yeah, we have carbohydrates, but not in very high extents. So in such cases, when in such questions, don't just say rice, bread. But they can be with the meeting. Right. We move to proteins. Well, we say proteins. What type of compound is it? Amino acids, yes, proteins, they build up of amino acids, right? It's, they have proteins in its structure. The building block of the protein is amino acids. One thing, proteins, very large compounds, complex molecules, like you suggested, it's made up of amino acids. And we need proteins about structure, function, and regulation of the body tissues and organs. So those are the functions. Building blocks of proteins, as you already said, amino acids. Speaking of amino acids, remember, right? Building blocks, amino acids, there are around 20 different amino acids in the nature. Well, they perform, they react together in different combinations to form all the proteins. Right? We have around 20, but by changing the combination, even if you say, think about two amino acids as A and B, right? So the proteins that protein option, what are the protein options which can be made by using the amino acid A and amino acid B as just symbolize AB, 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 one protein. AAB, 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 another protein. Right? ABA, 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 likewise, you see, uncountable number of different proteins can be made just with two amino acids. So when we have 20 different amino acids in the nature, naturally, obviously, we can make a load of proteins. Because we know that we need a lot of proteins because all the enzymes also are proteins, right? Then we have a number of thousands of chemical reactions in the body. Then we need thousands of enzymes also. Then thousands of different proteins should be there. All those are made by these 20 different amino acids. When it comes to amino acids, some are essential amino acids. මතකද මොනවාද essential amino acids කියලා කියන්නේ? අය කියන්නේ අන්න බොඩි එකේ නිබඳ ගන්න බැරි ඒවා. බොඩි එකේ නිබඳ ගන්න පුළුවන් නම් ඒවා non essential. ඒ කියන්නේ ඕන නැති නෙමෙයි. ඕනි but අපිට ඇඟ ඇතුලේ හදා ගන්න පුළුවන්. ඒවා non essential. ඒතකොට essential කියන්නේ මොකක්ද නෙමෙයි. Definitely we have to take it from outside. Right? එතකොට කියන්නේ ෆුල්ම vegetarians ලාට ඒක හම්බ වෙන්නේ නැහැ. එතකොට ෆුල්ම vegetarian නම් අපේ vegetarian කවුරු හරි ඉන්නවද? නැහැ. ඔක්කොම මොකද කරන්නේ? Good. right? Full vegetarian night. But it is very highly advisable to take those amino acids as supplements, right? That we have get from pharmacy. Essential amino acids, they cannot be produced in the body, must be taken from outside. അങ്ങനെ amino acid structure ഒക്കെ ഉണ്ട് അതിന്റെ carbon atom එකක් තියෙනවා ඒක වලේට group හතරක් තියෙනවා ඒක side එකක් C level වල අනිත් side එකේ H2 එකක් hydrogen අනිත් එක R group එක ඒ R group එක තමයි වෙනස් වෙන්නේ from amino acid to another amino acid ඒතකොට 20 different structures can be different R group එක වෙනස් වෙනවා right. Functions wise, what do the proteins do in the body? Structure. Governance. Energy. Energy. Take carbohydrates. Oxygen. Hemoglobin. Enzymes. About muscles, right? Muscles building and repairing tissues. Muscles. Enzymes and hormones. Hormones of hormones, proteins නෙමෙයි. සමහර hormones enzymes නම් ඔක්කොම protein. හැබැයි සමහර hormones protein නොවන ඒවා තියෙනවා. And supporting immune function, right? And transporting molecules. That's what you suggested. Hemoglobin carries oxygen. And serving as energy source when needed. Americium enemy when needed. Right. High protein foods. Meat, fish, eggs. And nuts and seeds are also soy products. So they are high protein structures. What happens if the body gets too little protein? They have protein and What are the problems that we have to face? Drop. Oh, yes. Enzyme activity is drop going on. Muscles growth අඩු වෙනවා. ඒකනේ දැන් බොඩි බිල්ඩින් කරද්දී අපි කන ප්‍රෝටීන් ප්‍රමාණය මදි නම් ඒකනේ වෙනම ප්‍රෝටීන් අර කීන්ස් කියලා අරගෙන ගන්න in right. So then that is like when the protein intake is not enough. දැන් ඒ ප්‍රෝටීන් අපි වැඩි කරගත්තාම ඒ ගන්න අවස්ථාවකදී එතකොට ප්‍රෝටීන් අඩුවෙන් ගත්තාම muscles grow වෙන්නේ right. And also immune system also is highly affected. And if we talk about the low protein intake, this situation for a child, a small one, then the growth rate is going to be low, right? There'll be delayed growth. Those are the problems. Well, about protein. So when we eat protein, now carbohydrates, we know, carbohydrates, fat, they can be stored in the body. Even if the මහත්තුනාට පස්සේ අපි ජීවිතලා ලස්සන්නේ ඇඟ ඒ කියන්නේ store වෙන carbohydrates, fat. It's store වෙනවා. So the විදිහට protein store කරලා තියාගන්න පුළුවන්. Okay. Proteins ඒක store කරලා තියාගන්න බෑ. ඒ කියන්නේ අපි දැන් muscle growth එකට ගන්න protein ඒ store කිරිල්ලක් විදිහට ගන්න බෑ. හරිද? දැන් muscles ගත්තට muscle එකක් කියන්නේ ඇඟේ පාවිච්චි කරන එකක්. හරිද? එතකොට ඒක muscle එක ඒක storage unit එකක් විදිහට consider කරන්න බෑ. හරිද? So ඒ කියන්නේ ඒක organ. So proteins අපි ඒක store කරන්න පුළුවන් එකක් නෙමෙයි. Body does not store protein. Any excess of protein is broken down and either used for energy or stored as fat. ඒක අපිට ආපහු metabolism මොනවද? Right? Protein විදිහට use කරන්න බෑ. තියාගන්න බෑ. ඉක්මනට fat වලට convert කරලා හැබැයි තවදුරටත් protein එකක් නෙමෙයි. නේද? තවදුරටත් protein එකක් නෙමෙයි. It's a fat. Because it is a result of a chemical reaction, not reversed again. Right. What's the role of enzymes and how are they related to proteins? Right. Substrate. Yes. And all the enzymes are proteins. Basically, finally, proteins are the ones those are doing that digestion job. Yeah. not only digestion, it's about all the chemical reactions in the body, right? So when it's related to food, it is digestion. And when it comes to the other reactions, it's minerals. The both are being done by enzymes. And all those enzymes are proteins. Right. Now we come to lipids. What can you remember about lipids? Come out of? Yeah. Glycerides, oil, and we were talking about cholesterol just before now in that test. So cholesterol, those stuff. Right. Are they polymers? No. Now, carbohydrates, yes, they are polymers. Proteins, they are polymers. But when it's lipids, no, it's not like that. It's a molecule. They are very compounds, like he said, fat, oil, waxes, steroids. Totally insoluble in water. Lipids don't dissolve in water. And this is also a source of energy and also insulation for the body. But lipids, what are the types of lipids that you remember? Hmm? Sorry. Triglycerides, oil, fat. Triglycerides. So fats and oils, they are coming under that category. And we have phospholipids. Phospholipids are there in the cell membrane. In the animal cell, right? In cell membrane, we have phospholipids. And we have steroids, cholesterol, hormones, right? ඔව් ඒකෙදී දැන් ෆ්‍රාන්සිස් කියන්නේ මොකක්ද? ඒකෙදී එක පැත්තකද මේ චේන් එකේ arrange වෙලා තියෙන්නේ දෙපැත්තකද කියලා. ඒතකොට දෙකම අයිති වෙන්නේ අර කැටගරියටම තමයි. ට්‍රයිග්ලිසරයිඩ් වලටම තමයි. ඒ කියන්නේ ෆැටි ඇසිඩ් එක ගත්තට පස්සේ ඒ කියන්නේ අපි ෆැටි ඇසිඩ් කියන එකේ ටයිප්ස් වලට ගියාට පස්සේ අන්න ඒක ගන්න පුළුවන්. ට්‍රාන්ස් එකයි සිස් එකයි. groups double bond එකේ එක පැත්තේද දෙපැත්තේද cis නම් එක පැත්තේ දෙපැත්තේ නම් trans. What do the lipids do in the body? Salbay, kalau the immunity kayak recommended salbay, kalau tadi itu mah asal terus. Bagaimana ya? Saat di bulan pas sedih paksiran, karena ini cukup ini kedua kita terus tidak boleh ada ini. Iya, kita kita udah kelas kita ada kelas gimana ini ya? Ada ඊට පළවෙනි වඩා පල්ලෙහි පිට ඔක්කොගේම ලකුණු එකතු කරාට පස්සේ පළවෙනියට වඩා වැඩි වෙනවා එකක්. හරි. මොනවාද මතක? What do the lipids do in the body? The temperature basically මොකද කියලා කියන්නේ? Insulation. Insulation. Right? ඒ කියන්නේ දැන් අපි සීතල පැත්තට යනවා කියලා. අපි හුඟක්ම කෙට්ටු නම් අරකට ටෝලරේට් කරගන්න අමාරුයි. හැබැයි මහත නම් අන්න ෆැට් ලේයර් එක තියෙන නිසා අන්න ඒක ඉන්සියුලේෂන් පුළුවන්. සර්වයිව් වෙන්න පුළුවන්. ඔය ඩිෆරන්ස් එක තියෙනවා. එතකොට ඒ ඉන්සියුලේෂන් ෆන්ක්ෂන් එක කරන්නේ ලිපිඩ්ස් වලින්. ෆැට් වලින්. right. Okay. Now another. Energy source, energy. Yeah. And yeah, what else you remember? And remember, when it comes to vitamins and stuff like, you know, only some vitamins are water soluble. They can be transported through blood, but some vitamins are not water soluble. They are lipid soluble, fat soluble, right? In that case, transporting those ones, that is important. And we need phospholipids to make the cell membrane. And as hormones, these are the functions of well, you pointed about cis and trans. Well, so at the first place, what do you mean by trans fats and why are they harmful to the body? Yeah. Yeah, related to cholesterol side, no? Yes. Trans. Right. Artificially created fats that raise bad cholesterol like he said, LDL, right, over the HDL side, increasing the risk of heart disease. Right, so the trans one is a heart disease related harmful substance. Well, cholesterol, definitely you should remember this because we did the test, cholesterol test. We are all about cholesterol test today, diabetes test. So, what exactly is cholesterol? Right. One point is like it's a type of lipid, and the steroid. We find it in animal cells, and functions wise, that is important for making hormones and cell membrane. Important in making cell membrane. That means it is having the structural design part. So that is why this is getting deposited on the inner wall of the veins and arteries, blocking the veins and arteries, which increases the risk of heart diseases. Right. Then when it comes to fat, we have to take fat from healthy sources. So what are the food sources where we get healthy fat? Yes. Yeah. Avocado, nuts, seed, olive oil, right? Fatty fish. Okay. Now we come to the reaction side of it. Metabolism. What do you mean? Sorry. Breathing energy. Is a result of metabolism. What exactly the direct idea about metabolism? Metabolism. It's about entirely all the chemical reactions in the body, right? It converts into energy and other necessary substances. Basically, in metabolism, energy is made, energy is created. And also in metabolism, other substances, the chemicals, the other substances are made. So the total of this is what we call metabolism. Right. Two main types of metabolism. Tinggal matikan ini. In respiration types like aerobic respiration, anaerobic respiration. Metabolism. Catabolism and anabolism. That is making substances and breaking substances. Right. What factors affect metabolism? That is all these chemical reactions which is happening in the body. Heat, enzymes, finally, yes, those factors, right, related to this. Because now, obviously, how fast the chemical reactions in the body, how fast the substances are being made, how fast the energy is being produced, how energetic we are, all depends on like how old we are, whether it's male or female, right? Muscle mass, physical activity, hormones, and the food that we eat, right? So all these will decide uh, about how much of energy that we have to run the metabolism, how much energy we get from the metabolism, right? So these things will change. Right. The role of enzymes in metabolism. What do you suggest? Other guy, oh, and then enzymes, right? So basically, enzymes only gather the reactants together. Basically, enzyme substrate complex is going to be formed, which we talked here under metabolism. We talked about that under digestion and stuff, right? Uh, then finally, it is decreasing the activation energy for the reaction. Basically, under a lower supply of energy, the reaction is happening. So in general, the catalyst speed up the reaction. Why? For any chemical reaction to happen, definitely there is some amount of energy required. Then what is happening? So this decreases the amount of energy, right? This decreases the amount of energy required for the reaction. So the rate of the reaction is going to be faster. Then in the presence of an enzyme, a particular given reaction will happen at a faster rate. Right. Okay. That's pretty much. So I ran through that revision to give you an idea that to show that what's the nature of the question is going to be, right? It is not hard at all, right? So get yourself prepared properly, right? You will get like around 2 weeks to study. I will confirm very soon. Because in July, you are starting the next semester, so right. So then before that, the exams will be done and results will be out. Results will be released. Are the other subjects exams scheduled already? Okay, no. Kalati, ne? SK1. It's 2 weeks. Doing somewhere near the what? 22nd. I will. That's all fancy. June 1st can cover this. June 1st වෙනකොට තියෙන 1 2 3 4 5 6 7 8 9 10 ආයෙත් 10 days තියෙනවා හැබැයි නේ. අවනේ. days extra කරන්නේ. එන්නනේ. And then hormones එන්නේ. මම පැහැදිලිව කියලා තියෙන hormones එන්නේ. එතකොට ඉතින් තව ඔට්ටල් තියෙන බාරක්. And then we'll get ready for the exam. And while you are studying, also whenever you get it out, quickly keep me posted, right? Then I will explain how it's happening and all, right? So study very well and get ready. Right, okay then. We shall wait for the exam. Study well. Yeah. Well, really? Yeah. Thank you everyone for the thought. Thank you. Certainly. Okay. Certainly. Certainly. We'll keep in touch, okay? Everyone. Certainly. 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 Certainly.